Good afternoon to lecturers. My name is Kevin, the group leader for the Wonders. Today, we are hosting a journal club discussion for an article entitled Quality of Whole Genome Sequence from Blood versus Saliva Derived DNA in Cardiac Patients. This article is published by BMC Medical Genomics on January 2020.
Firstly, we need to know what is whole genome sequencing. Whole genome sequencing is an important tool for detecting genomic variation. It is mostly used blood-derived DNA and can be used for both research and clinical application. The cost for whole genome sequencing decreased continuously nowadays. But other than blood-derived DNA, saliva-derived DNA can be used for error genotyping and whole exome sequencing also, as long as the quality of human DNA in each sample is sufficient. The advantages of using saliva-derived DNA for sequencing are the ability to get a sample non-invasively in participants who decline blood collection, the ability to mail saliva collection kit to remote location without requiring participants to visit a phlebotomy center, the stability of saliva sample for years at room temperature without the need for immediate DNA extraction, and it is more preferable to be used for patients who exposed to blood transfusion or hematopoietic stem cell transplantation from an allogenic donor, or have leukopenia, which can decrease the DNA yield from blood. Other than advantages, the disadvantages of using saliva-derived DNA are lower DNA yield from saliva, high microbial contamination if saliva is not properly collected, and the inability of younger children to provide saliva as per instruction. So, this experiment was conducted to compare the quality and quantity of DNA for blood-derived sequencing and saliva-derived sequencing for whole genome sequencing.
The aim of the research is to compare the sequence coverage, the yield, and concordance of single nucleotide variants and copy number variants called between blood and saliva genome. To increase the usage of saliva that have higher sample stability for participants who decline blood collections. To aid WGS process for patients who have exposed to blood transfusions, stem cell transplantation, or have significant lower DNA yield in blood.
Method used in these experiments start with study samples. 531 participants for blood-derived DNA, 502 participants for saliva-derived DNA. 5 unrelated individuals participated in the biobank to compare for quality of WGS. Participants were not allowed to eat, drink, and smoke for 30 minutes before saliva collection. At least 2 milliliter saliva is needed for this experiment. Next is DNA quality. 2 to 5 milliliter blood collected in EDTA tubes, 2 to 4 milliliter saliva collected using Origin saliva kits. All samples undergo agarose gel electrophoresis and their quality and quantity was measured. 1 microgram DNA used for WGS. Next is sequencing, read alignment, and variant calling. Illumina HiSec X is used to run WGS. Several software such as Isaac Aligner, Isaac Variant Caller, Control Free C, and Canvas is used to get the result. Next is downsampling. Software such as SumTools is used to account for possible bias. All subsequent analyses on each pair of blood and saliva sequencing were performed in the complete datasets and in downsampled datasets.
Next, they created gene sets for cardiovascular disorder-associated gene CVD, canonical transcript for CVD genes, as well as for all genes by using CCDS database and ensemble as gene source. Microbial contamination analysis was done to check microbial contamination by comparing the proportion of remap reads to human genome versus microbiome. After that, they compared the sequence coverage between blood sample and saliva sample by using paired t-test on the percentage of coverage at more than or equal to 20 times. Lastly, they did a variant comparison between the blood and saliva to test for several types of variants such as short variant calls S and V and copy number variant C and V.
Now I would like to talk about the results shown in this article. There are 46% of saliva samples fill QC requirements for WGS, which is whole genome sequencing, while there are 6% of blood samples fail to meet the QC requirement. For the microbial contamination analysis, there are 95.5% of whole genome sequencing reads from blood samples, and 82.6% of saliva samples initially mapped to the HG19 human reference genome, and final proportion increased to 98.2% and 93.8% respectively. Read mapping HG19 human reference genome was effective in excluding most of the reads from the human oral microbiome. Saliva samples 5 show a high level of microbial contamination. Now I would like to talk about the coverage analysis. The mappable mean depth of the coverage was more than 30 times for all of the blood samples and 4 of the 5 saliva samples. Saliva samples has overall adequate depth of coverage, which was not significantly different from coverage in blood. The cumulative coverage in consensus CDS, CCDS transcript of all reads shown as below. Figure 2 shows that average sequencing depth curve displaying the cumulative proportions of the whole genome sequence covered at a minimum of 20 times sequencing depth for the entire sets of reads in 5 pair blood and saliva samples.
Now let's look at the variant used between blood and saliva. Based on the figure, we can see that all SMV concordance show no difference in the types or unique SMV between blood and saliva sample. Next, rare SMV concordance shown in the bar graph reveal that the blood and saliva genome was 90.4 plus minus 3% genome-wide, 93.2 plus minus 2% in all CCDS transcripts, and 93 plus minus 2% in CVD genes. Furthermore, in CMV concordance, there were considerable variability in CMV calling rates across both blood and saliva samples. Average number of CMV call were 14% lower in saliva compared to blood. Average proportion of CMV in blood and in saliva genome was lower at 76% genome-wide, 77% in all CCDS transcript, and 78% in CVD genes. Comparison for all CMV in each sample across the entire genome in CCDS transcript and CVD genes transcript, including shared and unique CMVs. Only CMV detection of saliva was lowest in sample 5, which is less than 60% compared to other 4 samples paired.
In this study, almost 50% of derived DNA has failed quality control for whole genome sequencing compared to blood-derived DNA. However, saliva genomes derived from high-quality DNA had good coverage, low microbial contamination, and a 90 to 95% concordance for sequence variant calls. There was a slight concern of microbial contamination in saliva-derived DNA because the results shown one of the saliva samples, which is sample 5, has higher proportion of reads that map to the human oral microbiome. This might be caused by improper saliva collection technique. So to overcome this problem, stringent quality control criteria to DNA quality should be applied and higher depth of sequencing can be used. In addition, we also found some of the articles have proved that proper technique for saliva collection and use of certain microbiome kit with antibacterial agents such as Origin kit may help to increase the quality and quantity of DNA. Therefore, based on the data, we conclude that blood remains the preferred source for DNA for whole genome sequencing, but high-quality saliva samples that meet stringent quality control criteria can be used for whole genome sequencing when blood-derived DNA is not available or is not suitable to be used.
For my comments, I would definitely agree that saliva is truly a great alternative for biofluid experiment. Firstly, saliva cytokine profile has been successfully used as biomarker of respiratory and immunology disorder in clinical diagnostics. Non-invasive saliva sampling improves compliance of individuals and allow multiple collections in one day without imposing too much discomfort. By observing the CG probes on both 450K bit strips and 70K platform, it shows almost identical number of differential mediated sites between PMBC and saliva. This means that the majority of the probes were significantly mediated in PMBCs. PBMCs, and salivas. And finally, the analysis of saliva simplified the assessments of DNA-mediated patterns in molecular epidemiology studies, and especially to increase compliance with vulnerable populations such as children.
I found this article from BMC Medical Genomics useful because this article shares similar results and point of views regarding our discussion of this topic. Overall, I think blood-derived DNA is still better than saliva-derived DNA for whole genome sequencing because there were concerns over reduced yield of DNA extracted from saliva, especially given the rigorous DNA requirements for high-throughput technologies. In this article, results shown total DNA yield is approximately twofold lower from saliva than from blood. The second reason is because of the evidence of greater protein contamination and DNA fragmentation in the saliva DNA, which affected the quality of DNA. In this study, saliva has a lower mean value of A260 to A280 absorbance ratio compared to blood extracted DNA, indicating more protein contamination in the saliva extracted DNA. Besides that, when comparing the DNA fragmentation, a more intense band of fragmented DNA is also visible in DNA extracted from saliva, even though both DNA types have a clear and high molecular weight band.
For my opinion, I think that saliva-derived DNA is better than blood-derived DNA. I agree that saliva-derived DNA can be an alternative way to conduct in whole genome sequencing because the collection process is user-friendly, painless, and cost-effective. It is more suitable for the patient who are afraid of pain, and the cost for conduct whole genome sequencing is lower. Besides, the purity of genomic DNA extracted from the saliva sample is not significantly different than that from the blood sample, and it is more preferable to be used for patients who are under treatment, which can decrease the DNA yield from blood. Lastly, the saliva sample is stable at room temperature for a long period of time, and the ability to collect saliva by using saliva collection kit. So, the patient can collect their saliva themselves by using saliva collection kit.
I stand with my opinion which blood will win against saliva. There are several reasons that I listed below based on the article that I referred. Firstly, variant detection, which included SNV and indels, were easily detected in blood. More novel SNVs found in saliva sample was majority is false, and it's suggesting here that it had false positive rates. Second, compared with the non-enriched saliva and buccal samples, CNV detection was more sensitive in blood-derived DNA. Lastly, blood is collected by a professional phlebotomist, leaving little risk of improper collection. For saliva sample, participants may provide too little material, especially when self-collected, or may ignore instructions to refrain from eating.
Based on the research article I found, I would agree that saliva wins blood. Saliva is a readily available specimen which can be collected by non-invasive procedure and contains many hormones, drugs, and antibody of interest in screening and diagnosis. Therefore, saliva levels are a more accurate reflection of the active hormone in the body, especially for steroid hormones, which are strongly bound in blood by specimens binding globulin. Not only that, saliva can be collected with device so that it will be stable at room temperature for extended periods. Moreover, saliva collection, when properly instructed, most individuals can collect adequate specimens without professional help.
There is also another type of blood sampling called dried blood sampling, DBS, that has been widely used in newborn screening. DBS also is a good candidate for WGS as it managed to result in a good exome target coverage without causing any loss during exome capture. Another advantage of using DBS is even though DBS cards are kept with any barriers between them, it brings no sign of cross-contamination. In this article, the researchers concluded that all DBS can provide sufficient amount of DNA to produce acceptable whole genome sequencing and whole exome sequencing data. Hence, I think I will go for blood as a better source for DNA in WGS.
Now I would like to talk about my comments about the comparisons of saliva and blood samples. In my opinion, I think that saliva is a good alternative way for blood in whole genome because It is more easily obtained and less invasive than blood or other tissue collection. Besides, it will greatly facilitate the detailed examinations of genetic variants that may be associated with specific traits or have experienced adaptive evolution. Moreover, there was no systematic bias of metagenomic information between exome capture and non-capture data of blood and saliva in this article. Africa's genetic diversities remain poorly understood, in part because many regions of the continent lack adequate healthcare infrastructure, which can make blood collection impractical and make saliva a better choice.
Now I'm going to do a simple comparison regarding the strength of blood and vividness of saliva samples in whole genome sequencing. As we all know, the use of blood-derived DNA is the current standard for whole genome sequencing, so there are many established procedures and infrastructure for biochemical and metabolites testing in blood. As for the vividness of DNA derived from saliva, this article highlighted two main problems. The first problem is oral microbiome contamination at the time of saliva collection. And the second one is copy number variance. CNV yield in saliva was lower than that in blood.
For the strength of saliva-derived DNA, the analysis of saliva simplified the assessment of DNA methylation patterns in molecular epidemiology study by increasing compliance with vulnerable populations such as children. And most individuals can collect adequate specimens with instruction without professional help. Other witness of blood-derived DNA are invasive process which requiring trained personnel to interact directly with the sample donor, and the lack adequate healthcare infrastructure in Africa make blood collection impractical.
For summary, as we know, blood is the best biofluid for research purposes, especially for whole genome sequencing. But what if blood is not available? After discussion, 4 out of 7 of us agreed that saliva is actually a great biofluid for whole genome sequence with condition that when it meets its quality criterion. So it comes to a conclusion that saliva is definitely a great choice for biofluid experiment when blood is not available.